Vitamin D transport protein is a multifunctional plasma protein with many important functions. These include transport of vitamin D, binding of fatty acids, removing toxins, and a range of less defined roles in modulating immune and inflammatory responses. Vitamin D transport protein occurs in all higher order animals and is produced in the mother's first milk to help boost the immunity of their newborns. Other sources of vitamin D transport protein include human and animal blood, bacular expression, laboratory synthesis. Vitamin D transport protein contains 3 domains, each with a diverse and important function. Domain 1. Vitamin D binding. Every cell in our body has a vitamin D receptor, and every cell in our body secretes a vitamin D hormone. Vitamin D transport protein binds to vitamin D already stored in the body and transports it to where it's most required. Domain 2. Fatty acid binding. The human body requires fatty acids for many vital functions, from building healthy cells to maintaining brain and nerve function. Our bodies can't produce fatty acids, so the only source we have is from our food. Vitamin D transport protein binds to these essential fatty acids and transports them to help our bodies heal and repair. Domain 3. Actin scavenger. The third domain on vitamin D transport protein is an important protein of the plasma actin scavenger system and operates like a refuse collector and transports toxins to the liver so that they can be eliminated from the body. On this third domain, we find 3 sugars required to activate our immune cells. Our white plasma cells are produced in our liver, lymph nodes, and bone marrow. From here they enter the bloodstream. However, at this time they are not fully active immune cells. Instead, they are dormant and consume just enough energy to stay alive. To become active immune cells, our plasma cells require vitamin D transport protein. Here we see our white plasma cells interacting with the 3 sugars on vitamin D transport protein to become fully active immune cells. So we have just seen how, in a healthy body, adequate levels of vitamin D and vitamin D transport protein help to maintain and develop immunity. So when faced with chronic diseases, our immune cells stay inactive. Our immune system is suppressed or totally collapsed. This is due to a little-known enzyme called Nagalase that is produced by cancers and most pathogens. Until Dr. Yamamoto researched and published Nagalase, we had no idea as to the actual cause of the immune shutdown that allows cancers and pathogens to thrive unchecked. What is Nagalase? Nagalase occurs naturally in the intestines, where it breaks down glycoprotein in our food. But Nagalase is also produced in quantities by cancers, viruses and bacteria to break down a specific glycoprotein in our blood, vitamin D transport protein. Nagalase cuts the vitamin D transport protein at the 3 sugars required to activate our immune cells. This leads to immune suppression or collapse. Unmolested by our immune system, cancers and pathogens can now grow unchecked. Because Nagalase is an enzyme, a catalyst, it performs this malicious ritual over and over again, and each time it comes away unscathed and unchanged. Thus, one Nagalase molecule can destroy a huge quantity of vitamin D transport protein molecules. Nagalase is a very sensitive marker and can indicate cancer growth before any can be seen during current scans or tests. Nagalase levels are directly proportional to cancer's or pathogenic load and can therefore be used to track the success of any intervention. Similarly, Nagalase testing can be used to monitor the potential return of cancer following treatment. Unfortunately, Nagalase is not widely known or accepted blood test and only available in a few countries. Why is this? There are many published research papers that confirm the science is correct. It may be simply that detecting a cancer not yet visible could lead to confusion in mainstream medicine. How can they know how to treat cancer if A. they do not know what type of cancer it is, and B. where the cancer is growing? The answer can be quite simple. Cancer, if detected early enough, can be eradicated by the body's own defenses, and this can be achieved by dietary changes, healthier living, and immune-boosting protocols. So, in summary, Dr. Yamamoto discovered that cancer cells, viruses, bacteria, and most pathogens secrete the enzyme called Nagalase. Nagalase sabotages the vitamin D pathways, so our immune cells remain indolent and our immune system is compromised in a danger of collapsing completely. This discovery, given more time and research, could have revolutionized our lives by detecting health issues before they manifest themselves as a chronic disease. Not only could millions of lives be saved every year, but billions would be saved in healthcare services. This discovery did not go widely reported. His next discovery received little publicity either. Fooling Nagalase and restoring health. Let's look again at immune cell activation. 3 different types of plasma cells upregulate to natural killer cells, B cells, and macrophages. Nagalase locks onto and cuts the 3 sugars from the vitamin D transport protein, which then float off into the cellular soup to be recycled eventually. What Dr. Yamamoto discovered next could and should have earned him worldwide recognition. Nagalase only attacked when 3 sugars were present. In his laboratory and using his own enzymes, he cleaved 2 of the 3 sugars off the protein, leaving the remaining sugar that activated plasma cells into macrophages. Dr. Yamamoto called his discovery GcMAF, glyco-macrophage activating factor. Would it now be possible to restore immunity in the face of chronic disease? For his cancer studies, Dr. Yamamoto took 30 patients who had already received conventional treatment of chemotherapy, radiotherapy, and surgery. Blood tests showed rising Nagalase levels in this group, which suggested cancer was again growing in the body. This is stage 4 cancer, and is less than 2% survive longer than 5 years. Administering 100 nanograms weekly intramuscular injections of GcMAF, all the patients' Nagalase levels returned to normal within 6 months. Follow-up tests showed normal levels of Nagalase and no return of cancers after more than 5 years. Dr. Yamamoto then ran a similar trial with similar results, but this time with 30 HIV patients. All began the trial with elevated Nagalase levels, which were normalized within 6 months by administering 100 nanograms weekly intramuscular injections of GcMAF. Follow-up tests years after showed Nagalase levels remained in the range considered normal. Dr. Yamamoto published 4 papers on the therapeutic usage of GcMAF in humans, and in all papers he can be quoted as saying, GcMAF is the most potent macrophage activating factor known to man, and yet it causes no side effects in humans. It is not possible to patent a naturally occurring protein, and so Dr. Yamamoto's discoveries were overlooked by the pharmaceutical industry. However, during the last 5 years, a few companies have appeared selling GcMAF online. These companies are often targeted and closed down by the authorities that are protecting the so-called public health. Recently, diluted versions of GcMAF have been used successfully as a protocol to help heal the symptoms that are labelled autism. Figures are difficult to come by, but it is undeniable that thousands of children have improved.